Welcome everyone, and thank you for being here today. I'm excited to introduce our topic, real-time quality assurance in sterile manufacturing. On this first slide, you'll see the title and my name, Dr. S.K. Singh. We'll be exploring innovative approaches to ensure quality and safety in sterile production through rapid microbial assessment techniques. Over the course of this presentation, we'll discuss the latest methods, their benefits, and how they can transform quality control processes. Let's get started. Traditional sterility tests typically require around 14 days of incubation, which means we have to wait quite a long time before we get results. This delay can significantly impact the release of batches and, ultimately, our supply chain, causing delays in getting products to patients who need them. Regulatory bodies are actually encouraging us to adopt more science-driven, risk-based approaches to microbiological testing, like the use of rapid microbial methods, or RMM. These methods are designed to be faster, more reliable, and more sensitive when it comes to detecting microbes. By implementing RMM, we can significantly reduce testing time, which helps streamline our processes and improve overall efficiency. It's a perfect example of how embracing innovation and scientific progress can benefit not just compliance, but also the supply chain and patient safety. So today, we'll explore how these new methods are transforming traditional sterility testing and what that means for our industry as a whole. Rapid microbial methods, or RMM, are innovative technologies designed to improve how we detect microorganisms. One of their key advantages is that they can identify these microorganisms much faster than traditional culture methods, which often take days. This speed is crucial in making timely decisions that can impact product quality and safety. In addition to speed, RMM provide both quantitative and qualitative data. That means they don't just tell us if microbes are present or not, but also how many there are, giving us a clearer picture of the microbial environment. This detailed information supports real-time decision-making, allowing us to respond quickly to any issues as they arise. Another important aspect is that RMM help reduce subjectivity in testing. Traditional methods can sometimes be influenced by human interpretation, but these digital technologies enhance data integrity and consistency. Overall, rapid microbial methods are transforming our approach to microbial testing by making it faster, more accurate, and more reliable, key factors in maintaining high standards of quality and safety in our processes. Let's talk about some of the key benefits of implementing rapid microbial methods, or RMM. First, we see faster batch release, which means we can get products to market more quickly without compromising quality. This is crucial for staying competitive and meeting customer demands. Next, enhanced product safety is a major advantage. RMM allows for more sensitive and accurate detection of microbes, reducing the risk of contamination and ensuring that our products are safe for consumers. Increased process understanding is another benefit. By using rapid methods, we gain deeper insights into our manufacturing processes, helping us identify areas for improvement and maintain consistent quality. Reduced manufacturing hold time is also important. Shorter testing times mean less downtime, which keeps the production lines running smoothly and efficiently. Supporting continuous manufacturing is a key driver here, as RMM aligns perfectly with that approach, enabling real-time quality monitoring and control. Finally, RMM supports compliance with major regulatory frameworks, such as the EU Annex I, FDA PAT, and Quality by Design principles. This alignment not only helps us meet regulatory expectations, but also fosters a culture of quality and innovation within our organization. Let's take a closer look at some of the key types of rapid microbial methods used today. First up, ATP bioluminescence, which is a quick way to measure microbial activity by detecting ATP, the energy molecule found in all living cells. It provides rapid results and is useful for assessing cleanliness and contamination. Next, flow cytometry, a powerful technique that sorts and counts cells individually using lasers. It allows for detailed analysis of microbial populations in a matter of minutes, making it highly valuable for real-time monitoring. Then we have laser-induced fluorescence, or LIF. This method uses lasers to excite fluorescent markers attached to microbes, enabling highly sensitive detection and identification of specific microorganisms. Moving on to molecular methods like PCR, qPCR, and RT-PCR. These are incredibly precise techniques that amplify specific DNA or RNA sequences, allowing us to detect even tiny amounts of microbial genetic material quickly and accurately, which is essential for pathogen identification. Solid phase cytometry combines aspects of microscopy and flow cytometry, scanning slides for microbes with high sensitivity and speed, especially useful for low-level contamination detection. Microcalorimetry measures the heat produced by microbial metabolic activity. It's a direct way to assess microbial presence and activity without the need for labels or dyes. Biosensors and microfluidic systems integrate biological detection elements with miniaturized devices, enabling rapid, on-site testing with small sample volumes, ideal for point-of-care applications. Finally, automated colony counters and AI detection systems enhance traditional culture methods by automating colony counting and identifying colonies through artificial intelligence, significantly speeding up the process and reducing human error. All these methods represent the innovative approaches we're using to make microbial detection faster, more accurate, and more adaptable to various settings. They really connect to the overall goal of improving food safety, clinical diagnostics, and environmental monitoring through rapid detection technologies. Let's take a closer look at some of the key techniques used in microbial detection. First up is ATP bioluminescence. This method detects intracellular ATP, which is essentially the energy source inside living cells. Because of this, results can be available in just a few minutes to a few hours, making it really useful for quick surface and water monitoring. Next, we have flow cytometry. This technique detects single microbial cells by analyzing how they scatter light. It's particularly valuable for assessing bioburden and water for injection, or WFI, monitoring, providing detailed cell-level information. Then there's PCR-based methods. These are highly specific because they detect microbial DNA or RNA. They're especially useful when you're trying to identify particular pathogens or analyze raw materials. However, one limitation to keep in mind is that PCR detects genetic material regardless of whether the microbes are alive or dead, so it doesn't always tell you about viability. Finally, solid phase cytometry, or SPC, offers very high sensitivity, detecting as low as 1 colony-forming unit per 500 milliliters. This makes it ideal for sterile manufacturing environments and water system monitoring, where detecting even tiny amounts of contamination is crucial. To sum up, each of these techniques offers unique advantages, and choosing the right one depends on your specific monitoring needs and the nature of what you're testing. Let's talk about some of the key benefits of real-time monitoring and microbial detection, or RMM. One of the main advantages is its ability to provide real-time or near real-time data. This means we can detect microbial contamination much faster than traditional culture methods. As a result, we gain higher sensitivity, which allows us to identify issues that might otherwise go unnoticed. This leads to better contamination control and a significant reduction in risks. Additionally, RMM helps us minimize false negatives and manual errors, which are common challenges with conventional testing. This automation and precision improve our overall process reliability. RMM also supports essential areas like environmental monitoring, water quality checks, and automated process support, making sure everything is running smoothly. Most importantly, having this kind of proactive, real-time insight enhances our facility's readiness for regulatory audits. It demonstrates our commitment to maintaining high standards and being prepared at all times. Overall, these benefits help us stay ahead, ensure safety, and improve operational efficiency across the board. Let's talk about some of the critical testing and monitoring processes in pharmaceutical manufacturing. First, sterility testing is essential to ensure that our products are free from any microbial contamination before they reach patients. Alongside that, bioburden testing helps us quantify the microbial load on raw materials, equipment, or final products, giving us a clearer picture of contamination risks. Environmental monitoring, which includes air and surface testing, is vital for maintaining a clean manufacturing environment and preventing contamination from the surroundings. Water system monitoring, particularly for purified water and WFI, ensures that the water used in production remains sterile and uncontaminated, which is crucial because water is a common vector for microbial growth. In-process testing allows us to check the quality at various stages during manufacturing, catching any issues early before the final product is completed. To enhance our aseptic practices, we implement aseptic process simulation, or APS, which helps validate our sterilization and aseptic techniques under realistic conditions. Real-time facility monitoring and trending give us continuous insight into environmental conditions, enabling us to quickly identify and respond to any deviations. All these testing and monitoring strategies are interconnected and form the backbone of quality assurance in pharma manufacturing, helping us ensure that every product is safe, effective, and compliant with regulatory standards. Supported by various guidelines and standards, such as the FDA PAT guidance, the EU GMP Annex 1 from 2023, which actually encourages the adoption of alternative rapid methods, as well as USB chapters 1223 and 1071. Don't forget pH. Your 516, which also plays a role here. The key requirements we need to focus on include ensuring method suitability and equivalency, making sure our methods are fit for purpose and comparable to traditional ones. Scientific justification is crucial. We need to be able to explain and support why we're choosing specific methods. A risk-based approach is fundamental, assessing and managing potential risks throughout the process. Lifecycle management and periodic verification are also essential to maintain method performance over time, ensuring continued reliability. From a regulatory perspective, these points align with what authorities expect from us. Overall, this framework emphasizes that adopting rapid methods isn't just about speed. It's about ensuring quality, compliance, and scientific integrity every step of the way. Let's now talk about some of the key challenges we face in method development and validation. First, there's the issue of high investment and the need for skilled personnel. Developing and validating robust methods requires significant resources and expertise. Regulatory acceptance can also be a hurdle, as it often varies depending on the method used, which means we need to stay adaptable and ensure compliance across different regions and standards. Data integrity and proper lifecycle documentation are critical to maintain trust and traceability throughout the process. We also need to focus on accuracy, precision, and specificity to ensure our methods reliably measure what they're supposed to. Linearity, limits of detection, and quantitation are other vital parameters that define the method's performance scope. Robustness and ruggedness are essential to confirm that our methods perform consistently under varied conditions. When comparing new methods to established, compendial ones, method equivalency becomes a key consideration. Routine performance monitoring helps us catch any deviations early, maintaining quality over time. Additionally, assessing potential matrix interference is crucial to ensure our methods are specific enough to give us clear, uninterfered results. Finally, there are specific validation requirements that must be met to demonstrate our method's reliability and suitability for its intended purpose. All these challenges highlight the importance of a thorough, well-planned approach to method development and validation, ultimately supporting the overall quality and compliance of our products. Let's take a closer look at the validation parameters we need to consider. First up is accuracy, which is all about how close our measurements are to the true value. It's essential because it tells us if our method is giving us correct results. Next is specificity, which refers to our method's ability to measure the analyte specifically without interference from other substances. This is particularly important in complex samples. Then we have precision, which indicates how consistent our results are when we repeat the measurement under the same conditions. Now, linearity is about how well our method's response correlates with the concentration of the analyte across a specified range. This helps us ensure our method can accurately quantify samples at different levels. Moving on, the limit of detection, or LOD, is the smallest amount of analyte that we can reliably detect, but not necessarily quantify. The limit of quantitation, or LOQ, is the smallest amount we can measure with acceptable accuracy and precision. Both are critical for understanding the sensitivity of our method. Robustness and ruggedness are also key. Robustness tests how well the method performs under small, deliberate variations in conditions, while ruggedness assesses its reliability across different operators, instruments, or labs. We also need to consider method equivalency versus classical methods, which involves demonstrating that our new method produces comparable results to established techniques. And of course, routine performance monitoring is vital to ensure ongoing reliability over time. Finally, all these validation requirements are foundational because they ensure our method is fit for purpose, reliable, and compliant with regulatory standards. They connect directly to our broader goal of establishing confidence in our analytical results and maintaining quality in our processes. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Feel free to contact us through the provided channels. Thank you so much for your time and attention today. We appreciate your engagement and look forward to assisting you further.